All right, here we are, Lab 6, Reproduction and Development. If we look at some of the objectives, first thing we're going to do is identify some anatomy in the male reproductive system and then the female reproductive system. We're going to look at some microscopic observations, specifically in the seminiferous tubules in the testes. We'll see where the sperm cells are being produced. And then again, we'll take a look, microscopic observation inside the ovaries. We'll look at some immature follicles, some mature follicles, and we'll identify some oocytes as well. I'm not sure how heavily we'll get into the stages of embryonic development in the lab because that's kind of model dependent, but we'll go a little bit into that. I will go over these functions with you. I will not label diagrams because the diagrams are available to you in this section over here, lab lecture notes and lab atlas. This lab atlas has all of those diagrams already labeled for you, so you could fill those in yourself. So let's start with some of these functions, beginning with the male. It's the scrotum that is a sac that stores the testes, right? And this sac is held outside of the body and helps to maintain a suitable temperature. It's the testes. This is called the male gonad. And these produce sperm cells. The epididymis, they sit up on top of the testes, and this is the site of sperm cell maturation and storage. They're stored there. They're maturing while they're stored there. The ductus deferens is the vas deferens, is a tube that delivers the sperm cells along with other secretions from the testes, from the epididymis, to the outside. These are going to be providing a blood supply. We have two glands, seminal vesicles, prostate vesicles. We'll just fill these in together. Fluids for the eye there in the form of semen. And if you go back to the notes, there are functions for the secretions coming from these two glands, helping with transport, neutralizing the acidity, the acidic environment. So they're providing alkaline solutions. This is a common passageway for not only these gametes, but also for urine. It's a urine pathway. And the urethra is inside of this structure. We could just say this is the organ. All right, moving on. Again, labeling. Here we have the seminal vesicles and prostate gland here, and the vas deferens goes up and around. The scrotum holds the testes. On top of the testes, we have the epididymis. And we see some arteries and veins supplying and draining blood from that area. And this is a sagittal view, exposing the walls of the urinary bladder. We could see the urethra comes down. It goes actually through the prostate gland, but the ductus deferens also hooks up through the prostate gland with the same urethra. So we could do the same thing that we just did in the males with the female, starting with the ovaries. This is the female gonad. And these are producing egg cells. So the oviduct is the site of fertilization. The uterus, we have the muscular layer in the form of the myometrium. And we have the inner layer in the form of the endometrium, which provides this site of embryonic attachment. The birth canal is separated from the uterus through this structure called the cervix. And then we have external structures. Here they're giving you hints, right? So they're showing you the walls of the uterus. Here's an ovary. Fallopian tube would go up and around. So the fallopian tubes are labeled on this side here. And then we have the vagina or birth canal here, along with external structures. Sagittal view, again, we have the thick, you can see the thick walls of the myometrium, ovary, fallopian tube, cervix, birth canal. One of the microscopic observations that we're going to do is going to be in the male and the testes in the form of seminiferous tubules. So this is, if you watch the lecture video already, you would know that seminiferous tubules are the site of meiosis, specifically spermatogenesis. Or sperm cell production. The opening of the tube is called the lumen, right? And this is where we find the sperm cells. The sperm cells, of course, are the male gametes. The interstitial cells are outside of the seminiferous tubules, and these are the specialized cells that produce testosterone. All right. So when we're trying to do this, we're going to be doing this. It says under low power. I'm not sure if that we're going to really be sticking with that, but let's see what this looks like in the lab atlas. Again, this is the lab atlas that is provided to you in Blackboard. And it labels all the diagrams. And here we see a cross-section of the testes, and it shows all the many, many different seminiferous tubules. So everywhere we see this clear coming through, this would represent the lumen of the tube. And then we see the boundary of the tube, the walls of the tube. So this is under the scanner view. We can't really see much on scanner view. We would have to zoom in. So that's a seminiferous tubule. That's another one. There's the lumen. Here's another one with a lumen, another lumen. The high power view, I think, is the best view. And it's probably the view that I'm going to ask you guys to go with. We see one large seminiferous tubule here. And we see the walls going all the way around. So this is one tube. Here we see the lumen. And you can see these cells in the lumen. You can see they have these long flagella. So these are early spermatids. These are immature spermatids floating around. We see them here. So going back, so this is what we're drawing, right? So going back, no, I'm going to discard that, to our lab. What do I need here? Well, I don't want to do the low power. I want to do the high power view that's provided in the notes in the lab atlas. So we're going to have an ocular lens of 10x. The objective lens for high power is 40x, making the total 400x. All right. So when you draw your seminiferous tubules in this space, draw it to scale as it appears on the in the picture. Draw it to scale. Label the seminiferous tubule. Label the interstitial cells outside the tubule. Label the cells inside the lumen. And what I would like you to do, because this distance across, right, from the first week, we established this distance across to be when we're under high power, 450 micrometers from here to here. When you draw your seminiferous tubules in the note section down here, what I would like you to do is estimate the diameter. Of the seminiferous tubule. Again, of course, your units are going to be in micrometers. All right. When we look at the female microscopic observation, this one might actually be on low power because we could see everything pretty good from low power. So under low power, the objective lens it doesn't change, right? It's still 10x, but this one is 10x. So the total magnification is going to be 100x. That being said, if we change from high power to low power, we're kind of zooming out. The distance across is going to be bigger, right? So from here to here, not as it's remember, there's an inverse relationship between field diameter and magnification. So as magnification decreases, the field diameter increases, right? So we're decreasing magnification, so we have to increase field diameter. So it's going to increase, actually. This is from week 1 when we established these. It's going to go from 450 micrometers. Now we're looking at 1,800 micrometers across. So what does that mean? Well, let's see what we're looking at, first of all. If we go back to the female, to this presentation here, and we go to the female models, which this is a uterus. I'm sorry, this is an ovary, excuse me. And you'll label those parts. This is what we're looking at here. Scanner view now. So this is a nice, mature Graphian follicle. You see a big antrum filled with fluid. We see the nice picture of the oocyte here. This looks like an egg cell that's ready to be released to the outside, right? All right, so this one is on low power now. So you could see we have developing immature follicles up here. And then, but yeah, we have this nice large mature Graafian follicle, big antrum, very visible egg. This is the view that I would like you to use. All right. If we continue through the lab atlas, you could see the different stages of development, the cleavage stages, the morula stage, the blastula stage. We could see inside the blastula or blastocyst, we have the inner cell mass forming along with the cells of the trophoblast. And then we see embryonic development. So you could independently look at these and use your books to identify these different structures. Now we see the umbilical cord, the amnion, the chorion. We see nice development of chorionic villi, the yolk sac, producing blood cells until bone marrow is present. We see the placenta, the amniotic sac, clear mucus plug, cervix. We could see, if we look through the umbilical cord, arteries, 2 arteries, 1 vein. All right, so that's that. Going back to the lab now. You will use that information to, that goes along with this, with these pages. You could label that. All right, so let me fill in whatever I can here. I do want to leave you with something to do independently, but we could start with these questions up here. Let me skip over questions up here. Nope, let's go back down. All right, let's start with these. So these first couple of questions are talking about the difference between diploid cells and haploid cells. Diploid cells are cells of the body, right? Where haploid cells are make reference to the number of chromosomes in the gametes. If you were to look at the testicular cells, well, the testicular cells are part of the body. So how many chromosomes are there? Those are diploid cells, 46. When we compare them to the actual sperm cells, well, those are gametes. So they're going to have half the number of chromosomes. They're going to have 23. What is the type of division that produces sperm cells? Well, this is a special type, right? It's called meiosis. And it's the seminiferous tubules that are producing these cells. Testosterone is coming from these interstitial cells. Remember from the notes in the book, we refer to interstitial cells as Leydig cells. And this influences the production of sperm cells, right? Influences or stimulates sperm cell production. It also is responsible for secondary characteristics, right? For males. All right, so looking at the ovary under the microscope, it's that follicle that carries the egg cell. The antrum fills with fluid as it develops into a Graafian follicle. But the mature ovarian follicle is still carrying the egg cell, the oocyte. So the oocyte is the female gamete. The corpus luteum is a structure that produces progesterone. Okay, so this is what's left over after ovulation. This is the actual, it's made from the walls of the Graafian follicle. It produces progesterone. However, if the egg cell does not become fertilized, the corpus luteum will degenerate as the macrophages of the white of the immune system begin to break this down, this structure gets filled with collagen. It becomes visible in a microscope. In fact, corpus albicans literally translates into white bodies, and you could see them. They're there. Okay, so these are structures that form as a result of the degenerated corpus luteum. All right, so here we see a Graafian follicle right at the site of ovulation, so the egg cells being released. We see a large fluid-filled antrum. Over here, we see smaller developing follicles. After ovulation, we see clear, nice corpus. Here's a corpus luteum. The corpus luteum would be releasing progesterone. If the egg cell does not become fertilized, the corpus luteum will break down, forming these white bodies called corpus albicans. The corpus luteum comes as a result of what's left over from the matured follicle after ovulation. So this differentiates into this. And then these break down into these. How many chromosomes would there be in the cells that make up the structure of the ovary? Well, the ovary is part of the body, right? So all body cells have 46 chromosomes. However, the egg cells themselves have 23 chromosomes. And they are made by a special cell division, just like in the males, called meiosis. The hormone that is secreted by a developing follicle is estrogen. What becomes of the follicle after ovulation? Luteum. Hormone secreted by progesterone. Okay, so estrogen and progesterone, those are our two uterine hormones, right? Hormones produced, ovarian hormones. Hormones are produced by the ovaries, but are in control or influencing the uterine cycle. So this builds the endometrium, which is the inner lining of the uterus, along with giving secondary characteristics as well. The progesterone maintains the thickness. When we compare sizes, the egg cell is much larger than sperm cells, maybe even 100 times larger. However, the egg cells are produced in such fewer numbers compared to sperm cells. Much fewer, by many, many millions. Egg cells that are produced somewhere in the neighborhood of 200,000, 400,000. Sperm cells produce somewhere in the neighborhood of 300 million per day. Two organs where meiosis is happening. Of course, that's the ovaries and testes, because meiosis is only used to form gametes. All right, so when you do this, you're going to use your field diameter for low power, 1800 micrometers, to estimate the diameter of the large Graafian follicle. Do we have that written up there? Oh, no, what happened? No, not that far. Sorry. So the mature follicle, remember, is the Graafian follicle. And it's labeled up there as a Graafian follicle. All right, again, your answer is always going to be in proper units, which is micrometers, just like the field diameter is in micrometers. So do not include this in your drawings. This is just trying to just show you. I don't want to see that in there. I just want to see your drawings and this in your notes. All right, and that's about it. Actually, I could help you with this too. When we look at the anatomy of the sperm cell, we could break it down into a head. The acrosome contains digestive enzymes. These digestive enzymes are critical in order to penetrate not only the cells of the corona radiata, but also that protein layer called the zona pellucida. So it can merge with the plasma membrane of the oocyte. The nucleus is going to contain the chromosomes, the genetic material. The midpiece is packed with mitochondria because remember, these cells are moving quite a bit, using a lot of energy. Mitochondria provide that energy as it is using this whip-like tail. So these are the male gametes. Remember that? Female gamete. Gamete are the egg cells, or the ova, or the oocyte. Any of those. Of course, this is a fertilized egg. So this is the product of sperm meeting the egg cell, and then we have mitosis happening. So after the first division of mitosis, we have what's called this 2-cell stage, and then we have the second division, which brings us to the 4-cell stage, followed by the morula stage, and then the blastula stage, and then the gastrula stage, which is featured by the presence of the 3 germ layers, including the ectoderm, the mesoderm, and the endoderm. Remember, these 3 germ layers are going to differentiate into all the tissues of the body. The placenta passes nutrients, oxygen, wastes between mom and baby's blood. Of course, the umbilical cord connects the embryo or the fetus to the placenta. All right. The yolk sac produces blood cells early before the presence of bone marrow. Once bone marrow is produced, then the blood cells will be coming from there. But before that's the job of the yolk sac. The amniotic sac protects the baby. Using its fluids. And you want to see the notes. There's a more detailed description of how the amniotic fluid helps to protect the baby. And of course, this is the site of development. All right. And this is where we're going to conclude. So you just have some labeling to do. You have some drawings you got to do. And then you're done.